Today we are going to study the regulation of glycolysis, and when I am saying regulation, that means which are the factors that can affect this glycolysis, and affect, that means whether it can be turned off or turned on. That means in which situations this glycolysis or the cells should perform glycolysis, and which are the factors that will control all this glycolysis process or this glycolysis of the cells, that may be of liver cells or muscle cells or adipose tissues, whatever it may be, right? So we will start from various the various inhibitor and activator of the glycolysis. In that, first you can observe this chart where in first column we have written the enzymes, in second column there are inhibitor, and in third column there are activators. Okay, so the very first enzyme which is involved in the glycolysis is nothing but the hexokinase. Okay, and now the inhibitor of this hexokinase is nothing but the glucose 6 phosphate. So glucose 6 phosphate is the itself, it is the inhibitor for the hexokinase. If the concentration of glucose 6 phosphate is more, then hexokinase can get inhibited. Then activator of this hexokinase, that means in which case it is going to show the activity of the that hexokinase is the AMP and ADP. That means, see, if the concentration of glucose 6 phosphate, that means the reaction which is catalyzing by the hexokinase is nothing but the conversion of glucose to glucose 6 phosphate. Okay, now if already there is the lot amount of glucose 6 phosphate present in the cell, then what is going to happen? Then this glucose 6 phosphate itself, it will get it will inhibit the activity of hexokinase, so that furthermore glucose 6 phosphate will not get generated. Similarly, in which condition it will catalyze the conversion of glucose to glucose 6 phosphate? When the concentration of AMP and ADP will be more, that means the lack of ATP. AMP and ADP concentration if more in the body or in the cell, that means this hexokinase has to now convert glucose into glucose 6 phosphate in order to perform the glycolysis. Secondly, PFK1, phosphofructokinase 1. Now this enzyme, we know that it is involved or it is catalyzing the regulatory step of the glycolysis, right? So when we are saying that it is catalyzing the regulatory step of glycolysis, that means it is the major enzyme which play the major role, or the main enzyme which play the major role in the glycolysis. And already we know that the activator of PFK1 is nothing but the fructose 2,6 biphosphate. Okay, now inhibitor of this PFK1 is nothing but the H plus ions, that means protons. If they are present in more amount, then it can inhibit the activity of PFK1. Then the higher concentration of ATP also. If already in cell ATP level is high, then or in the blood ATP level is high, then this higher ATP level can inhibit the activity of PFK1, phosphofructokinase 1. Then citrate is there. If concentration of citrate is more, then it can also inhibit the activity of PFK1. Next, what are the activator? One activator we know that fructose 2,6 biphosphate is the activator of PFK1, right? Then along with this fructose 2,6 biphosphate, AMP and ADP are also the activator of the PFK1. So AMP and ADP, see, whenever there will be the need of energy in the body, right? That means energy already has been get utilized by the cells, right? That means ATP has been getting dephosphorated and it can get converted into AMP and ADP. So the level of AMP and ADP will increase and it will then activate the activity of the PFK1, right? So it is going, they are going to activate the PFK1. Next, last enzyme is pyruvate kinase. Okay, this is also one very important enzyme in the glycolysis. Now, inhibitor for this is nothing but the ATP. If ATP concentration is higher, then there is no need to produce ATP, and that's why ATP will inhibit the activity of pyruvate kinase. Along with alanine, alanine is also the inhibitor of the pyruvate kinase, it can also inhibit the activity of pyruvate kinase. Then what are the activator? Activators are fructose 1,6 biphosphate. This is the activator of this pyruvate kinase enzyme. So these are the general inhibitor and activator of the some three major enzymes that we have seen or studied in the glycolysis. Now we will move, this is the type of allosteric regulation. Now we will move on to the hormonal regulation. Okay, we will move on to the hormonal regulation, how hormones are going to control or they are going to regulate the process of glycolysis. So in that very first situation we are going to consider is nothing but the when if the blood glucose level is low, that means in blood the glucose level is low. At that condition, what is going to happen? At that condition, alpha cells of the pancreas, they are going to secrete the glucagon hormone. Okay, the alpha cells of pancreas are going to secrete the glucagon hormone. Now all of us know that glucagon has to it has the role to play in the gluconeogenesis, because there is already low blood glucose level, right? So that liver has to pump the glucose in the blood, right, in order to run the glycolysis. But we are talking about the condition where blood glucose level is low. So in that condition, alpha cells of pancreas, they will secrete one hormone, and that hormone is nothing but the glucagon. Now see what is the exact mechanism of this glucagon. Okay, now the receptor for this glucagon, now because gluconeogenesis will get it will be occur in the liver. So the receptor for this glucagon, they are present on the liver. Okay, they are present on the liver cells. Okay, now what is going to happen? As soon as these alpha cells of pancreas will secrete the glucagon hormone, it will give the signal. That means secretion of this glucagon is the signal for the liver to proceed for the gluconeogenesis. Okay, but here we are talking about the glycolysis. What will be the effect on glycolysis? So if already blood glucose level is low, then it has to stop the that hormone has to stop the glycolysis process or glycogenesis process, where glycogen is going to form, right? So in what exact manner it is going to happen? See, as soon as glucagon will get released or secreted by the alpha cells of pancreas, then it will bind with the receptors that are the G protein receptor we say. For example, this is the liver cell. Here these receptors are present. Okay, so here glucagon is going to attach, right? This glucagon is going to attach to the liver cells where the receptor for glucagon are present, and these receptors are nothing but the GPCR, G protein linked receptors. Okay, so these receptors, they will identify this glucagon, then this will activate. As soon as this glucagon will attach on this receptor, this will activate G protein. Okay, because these are the receptors which are G protein coupled receptor. Okay, these GPCR are nothing but the G protein coupled receptor which are present on the liver, and they are going to attach, or glucagon is going to attach on these receptors. Okay, as soon as glucagon will attach to these receptors, what it will do? It will activate G protein. Okay, it is going to activate the G protein. Okay, now what is the effect or impact of this activation of G protein? This activation of G protein is going to activate the adenyl cyclase. Okay, adenyl cyclase will get activated, through which ATP will get converted into the cyclic AMP. Okay, see what is this sequence of event here. Okay, see, when already glucagon has given the signal. Okay, here glucagon is there which is giving signal, and this glucagon is going to bind on the GPCR receptor, and when it will bind on the GPCR receptor, this GPCR receptor, they will get signal and they will activate the G protein. So initially G protein is getting activated. Now this G protein, activation of G protein will lead to the activation of adenyl cyclase. Okay, it is going to activate the adenyl cyclase. Remember, this all is happening over inside the liver now. Okay, inside the liver only. So adenyl cyclase will get activated, and this adenyl cyclase, what it will do, it will convert the ATP into cyclic AMP. It will convert the ATP into cyclic AMP. Impact of this cyclic AMP is it will bind with the protein kinase, because see, it is ATP is getting converted into cyclic AMP, right? And that's why this inorganic protein which has been getting released here, it will bind with the protein kinase A, and when this inorganic phosphate which is released from this ATP, right, when it will bind with the protein kinase A, that is pKa, protein kinase A, when that inorganic phosphate is going to bind with this pKa, it is going to activate that pKa. Okay, it is going to activate that protein kinase A enzyme. Okay, so this is the sequence of event up to this. What will happen after the activation of pKa? Now once this pKa will get activated, then this activated pKa will phosphorylate the variety of substrate or variety of targets. Okay, this pKa has variety of targets, and as that name suggests, protein kinase A, right? So kinase, that means it is going to phosphorylate the target, whichever substrate it is going to phosphorylate that. Now this phosphorylation, for example, phosphorylation of say pyruvate kinase or you can say glucokinase, okay, glucokinase or pyruvate kinase. So this phosphorylation of these substrate, what it will do, it will inactivate them. It will inactivate that substrate. Okay, so inactivation will be there after the phosphorylation, and this phosphorylation is occurring due to the inorganic phosphate which has been released from this ATP, and that has been activated this protein kinase A. Okay, then this protein kinase A, it is going to now phosphorylate other targets which are present in the glycolysis. Okay, for example, it can phosphorylate pyruvate kinase, it can phosphorylate glucokinase, and it can also phosphorylate PFK2. Okay, and the regulatory step of the glycolysis is the formation of fructose 2,6 biphosphate. But before fructose 2,6 biphosphate, there is one intermediate, and that is nothing but the fructose 1,6 biphosphate, and that reaction is catalyzed by the PFK2. Okay, and if PFK2 is getting inactivated, that means this fructose 2,6 biphosphate, it will not get generated, it will not get formed, because the activator for this PFK1, which is now catalyzing the reaction of formation of fructose 2,6 biphosphate. The activator for PFK1 is the fructose 1,6 biphosphate, right? Fructose 2,6 biphosphate. So this fructose 2,6 biphosphate will not get synthesized if PFK2 is getting inhibited or is getting inactivated, and it can get inactivated when this pKa will phosphorylate that PFK2. Okay, what will be the exact impact of this, all of this now? This can turn off your glycolysis. Okay, so this total will turn off the glycolysis of that liver. Okay, now glycolysis has been turned off, right? Now along with this inactivation of certain substrates which are there in the glycolysis, this pKa can activate or inactivate glycogen synthase also. Okay, that means firstly it is shutting down the glycolysis, and secondly it is inactivating the glycogen synthase. That means it is now it has been now turn off the synthesis of glycogen also, because already in blood there is low sugar level. That's why glycogen synthesis will also get stopped, and this is the effect of pKa only, right? Now it is going to inactivate glycolysis and also glycogen synthase. but it is going to activate the glycogen breakdown, and that glycogen breakdown, all of us know what it means, glycogenolysis. Okay, because now this glycogen which has been stored, right, this glycogen which is stored in the liver, that has to be get breakdown in order to resynthesize glucose, in order to release the glucose in the blood, because there is already low blood glucose level is there, right? And that's why this glycogen will now get breakdown. This is also activated by the pKa. So it is shutting down the glycolysis, it is stopping the synthesis of the glycogen, but it is activating the breakdown or lysis of this glycogen molecule, and then gluconeogenesis will run. Okay, and that's why we say glucagon has the positive impact on gluconeogenesis and negative impact on glycolysis. Okay, right? So here is all this sequence of events which are take place in this hormonal regulation of glucagon. Okay, now one another point to be noted here, a very important point is that this all is happening, means gluconeogenesis will also run inside the liver and glucose will form inside the liver, but where we want that glucose, we want that glucose in the blood. Okay, so in order to transfer or transport this glucose from liver to the blood, there is one transporter, and that is nothing but the GLUT2 transporter. Okay, GLUT2, you can say GLUT2 transporter. Here I will write it. See, GLUT2. This transporter, glucose transporter, GLUT2 transporter, is involved in the transport of glucose from liver to blood. That means liver will release now the glucose inside the blood when gluconeogenesis will get completed. This is the impact of glucagon on the glycolysis. Okay, I think this must be clear to all of you now. Second, now in the second condition we are going to talk about the when the blood will contain high glucose level. That means when blood is having the high glucose level. Okay, so high blood glucose level, it will now stimulate the beta cells of pancreas, and beta cells of pancreas are going to secrete the hormone named insulin. Okay, so beta cells of pancreas at the high blood glucose level going to secrete insulin. Okay, after secretion of this insulin, again there will be this sequence of some events. Okay, and this sequence of events will then ultimately impact the glycolysis process of glycolysis. How that we are going to see now. So see, first insulin, first of all, the receptors for the insulin, they are majorly present on the liver cells, then adipose tissues, then muscle cells. Okay, so on that means your liver cells, muscle cells, then adipose tissues, on these the receptor for insulin are present majorly. Now the receptors of the insulin, they belong to the family RTK. Okay, RTK, they will belong to the family RTK, and this RTK stands for the receptor tyrosine kinase. Okay, receptor tyrosine kinase. Now this receptor of insulin, they belong to this receptor tyrosine kinase family, right? Then this keep in mind for further reference. Next, now as soon as these beta cells of pancreas will secrete insulin, it will give a signal, that is the signal for the cells now, okay, which are having the receptors of this insulin. So this signal will come, then this insulin is going to bind with the RTK, which are already present on the liver, muscle or adipose tissues. Okay, liver cell, muscle cells or adipose tissue, on their cell or tissues, these receptors RTK, that is your receptor tyrosine kinase, these receptors will be present. These will now identify this insulin, and that insulin will bind with that RTK. What will be the impact of this now? This will lead to the activation of phospholipase C gamma. Okay, PLC gamma, you can say PLC gamma, in short PLC gamma. So as soon as this insulin is binding with the RTK, receptor tyrosine kinase, which are present, again I am telling you, on the liver, muscle and the adipose tissues. Okay, liver cells, muscle cells and adipose tissues. So after this binding, it will activate the phospholipase C gamma, or you can say PLC gamma. Okay, this PLC gamma when get activated, it will activate the PIP2 kinase. Okay, PIP2 kinase enzyme will get activated, then it will form the PIP3. Okay, it will form PIP3, which will lead to activate the or formation of the IP3 and DAG molecule. Okay, this IP3 and DAG will then activate PKB. Okay, what are this we are going to learn? Okay, this IP3 or DAG, DAG means the diacylglycerol, IP3 is the inositol triphosphate. Okay, so these molecules will get generated after this signal has been come, after this insulin has been bind with the RTK, then this PLC gamma has been activated. It will activate PIP2 kinase, formation of PIP3 will be there. This PIP3 will lead to form IP3 and DAG molecules. This IP3 and DAG molecule will activate PKB as well as protein phosphatase. Okay, they will activate PKB as well as protein phosphatase. Okay, then this PKB which is activated now, okay, this activated PKB is going to phosphorylate the RAB and activate it. RAB is now all of you know, it is the monomeric G protein. It is monomeric G protein which is involved in the vesicular fusion. It is involved in the vesicular fusion because see, for the glucose transporter in the presence of this glucose, in the presence of this high blood glucose level, when insulin is getting secreted, then this glucose which is present, extra glucose which is present in the blood, that has to be get transported into the adipose tissues, and that adipose tissues must accept that glucose, and for that transport there is one transporter, glucose transporter, and that is nothing but the GLUT4. Okay, this is the only glucose transporter which is under control of the insulin. Okay, this GLUT4 transporter, it will transport that glucose to the adipose tissues, because adipose tissue we know, adipocyte with they are nothing but the fat storage body. Okay, so extra glucose after the formation of ATP, right? So the extra glucose which is remaining, that will get stored as the fat in the adipocyte. Okay, in the adipose tissues, and for that purpose this glucose, extra glucose, has to transfer, or it has to transported to the adipocytes, and that can be transported through the glucose transporter GLUT4, which is the only glucose transporter that is under control of insulin. Okay, so this PKB will phosphorylate this RAB molecule, which is the monomeric G protein which is involved in the vesicular formation, because these GLUT4 transporter are initially present on these vesicles. Okay, they are present on the vesicles, and as soon as this insulin or this PKB, this RAB PKB, activated PKB is phosphorylating RAB molecule, this RAB molecule is now they fuse this vesicle on the membrane of this adipose tissues. Okay, and on the adipose membrane of the adipose tissues, now the receptors, okay, this will increase, the number of receptor will increase on the adipose tissue membrane, and as these receptors will increase the number of, now they can identify that insulin, and they can now intake that glucose via the GLUT4 transporter. Okay, so this RAB will get activated, then vesicles which containing GLUT4 will fuse with the adipocyte membrane. They are going to fuse with the adipocyte membrane, which will increase the number of GLUT4 over the adipocyte membrane. And GLUT4 is nothing but the glucose transporter. It is going to transport the glucose from blood to adipocyte, and it is the only transporter which is under control of the, or receptor under control of the insulin. Okay, so in this way, this Now after this, what will happen, you can simply guess, means this glucose is going to get now converted into the glycogen, or it will get stored in the form of fat in the adipocyte. Okay, in liver it will get stored in the form of glycogen, glycogenosis. Okay, and in these adipocytes it will get stored in the form of fat. So this is the overall complete regulation of the glycolysis. I think you must understand now it, right? You can simply have the notes of this, and if you have any doubt, you can post me that doubt in chat box, right? Okay, thank you.